Hi, my name is Ankita Adhvaryu, and this summer I worked on hunting for phagelets, using a Mycobacterium smegmatis Lysogen to search for phage-like particles. A novel phage-like particle called a phagelet has recently been identified, which has been found to infect Mycobacterium achiense. Upon infection of M. achiense, it induces expression of the Herbert WM prophage, which is the only phage known to associate with this species. While phagelets appear similar to bacteriophages, they have several key differences, including their smaller genome, lack of tail proteins, and lack of a lysine cassette. As a result, it likely cannot successfully infect bacteria independently, like a normal bacteriophage or phage would. It's hypothesized that during egress, the phagelet borrows the prophage's tail proteins and lysine cassette. This particle has thus far only been observed in M. achiense, and while there are similar satellite phages in other hosts, these phagelets are quite unique. This project aims to address our current gaps in knowledge concerning phagelets by studying possible activity in another bacteria, Mycobacterium smegmatis, which is a common bacterial host used in laboratory studies and is especially of interest due to its use as a proxy for M. tuberculosis. The study aimed to address two primary research questions. Are phage-like particles found in M. smegmatis, and are phage-like particles a widespread phenomenon among the Mycobacterium genus? The first phase of the experiment involved creating a culture of M. smegmatis containing a bacteriophage. We primarily focused on using phage lysate samples isolated in previous years at William and Mary. First, a plate of host M. smegmatic cells was prepared, and a spot test was conducted using dilutions of the high-titer phage lysates. After several days of incubation, a turbid lawn had grown on the plate, and there were clearings where the lysate dilution had been spotted. The spots were checked for mesas, an overgrowth of bacterial cells in the zone of clearing. The mesa was then streaked for isolated colonies onto a new plate. Following incubation, isolated colonies from the plate were used to conduct a patch test using one plate with a lawn of normal enzymatis and another without. After incubation, if a halo was observed around the patch on the plate with the lawn, this indicated phage release and suggested that the colony tested was a good lysogen candidate. For each candidate, 3 successive rounds of purification were completed. Following the final purification step, the patch test was repeated to confirm continued lysis. Afterward, a liquid culture was created of the M. smegmatis Lysogen containing the corresponding phage. This culture was used to conduct an immunity assay, in which it was plated and dilutions of various high-titer phages, including the one used to create the putative lysogen, were spotted onto the plate. Post-incubation, if the original lysate used to create the culture did not cause any clearings, then the culture was considered homoimmune, and the protocol could proceed to the next phase. This process was repeated for each lysogeny culture created. In the second phase, the lysogen was used to screen for possible phage-like particles in the environment. First, a soil sample was collected, mainly focusing on muddy and damp locations. The soil was then enriched in liquid media with the lysogen culture and incubated overnight to promote viral growth. The resulting liquid was filtered extensively so that only phage would be left behind. Then it was allowed to infect liquid cultures of normal enzymatis and the lysogen, which would both be plated and incubated for several days. If the lysogen plate displayed circular clearings or plaques in the lawn, but no plaques were observed in the enzymatis plate, then this suggested that there may be a phage-like particle involved. The plaques were removed from the plates and placed into phage buffer for further dilutions and platings to isolate the plaques. Dozens of lysates were tested, but only a handful made it to the end of phase 1 still putative lysogen, including Doc McStuffins, Deb65, Marpesa, Ivyline, and Legote23. There are more in the process of being confirmed. As of now, no phage-like particles have been detected yet. However, research is ongoing, and environmental samples continue to be screened for any signs. Should any be found, the next step will be to sequence and annotate its genome to compare it to the phagelet previously identified in M. achiense with Herbert WM. This data would provide evidence of whether these phagelets are a widespread phenomenon in Mycobacterium and provide insight into evolutionary relationships. Furthermore, applications of phagelets and similar particles may open new doors in the field of phage therapy. Regardless of the results of the hunt, this study marks a significant step in the exploration of novel phage-like particles as a whole.